Mitochondria, structure, function, and oxidative phosphorylation. Structure of mitochondria. Mitochondria are known as the powerhouses of the cell because they generate most of the cell's supply of ATP, used as a source of chemical energy. Each component of the mitochondria plays a specific role in energy production. Morphology of mitochondria, shape and size, typically rod-shaped or oval-shaped, but the shape can vary. Size ranges between 0.5 to 1 micrometer in diameter and up to 10 micrometers in length. Components of mitochondria. A. Outer membrane, composed of 50% lipids and 50% proteins, contains large, protein-based channels called porins that make it permeable to ions and small molecules. Enzymes involved. Monoamine oxidase, fatty acid elongation enzymes. B. Inner membrane, rich in proteins, about 80% proteins. Contains enzymes of the electron transport chain, ETC, and ATP synthase, folded into cristae, which increases surface area for ATP production, highly impermeable to most molecules. Transport proteins are required. C. Intermembrane space, the space between the inner and outer membranes. Contains cytochrome C and enzymes for ATP/ADP exchange. D. Mitochondrial matrix, contains mitochondrial DNA, mtDNA, ribosomes, and enzymes of the Krebs cycle. Houses enzymes for the oxidation of pyruvate and fatty acids. Enzymes: pyruvate dehydrogenase, isocitrate dehydrogenase. Cristae are folds in the inner membrane that increase the surface area for electron transport and ATP synthesis. Cells with higher energy demands, e.g., muscle cells, have more densely packed cristae. Mitochondrial DNA, circular, double-stranded DNA, encodes some of the mitochondrial proteins. Most are nuclear encoded. Inherited maternally in most organisms. Functions of mitochondria. Energy production. Mitochondria perform oxidative phosphorylation to generate ATP. Metabolic role. Krebs cycle, citric acid cycle, occurs in the matrix. Beta oxidation of fatty acids occurs in mitochondria. Calcium homeostasis acts as a calcium ion reservoir, regulating intracellular calcium levels. Apoptosis, programmed cell death, releases cytochrome C to activate apoptotic pathways. Heat production. In brown fat, mitochondria generate heat. Non-shivering thermogenesis. Oxidative phosphorylation. Oxidative phosphorylation is the process where ATP is synthesized using the energy derived from electron transfer. 1. Location occurs in the inner mitochondrial membrane, specifically across the electron transport chain, ETC, and ATP synthase. 2. Stages of oxidative phosphorylation. Step 1. Electron transport chain, ETC. The ETC consists of 4 protein complexes, 1 to 4, and associated molecules. Complex 1. NADH dehydrogenase accepts electrons from NADH, transferring them to ubiquinone, COQ. Pumps 4 protons, H+, into the intermembrane space per electron pair. Complex 2, succinate dehydrogenase, oxidizes FADH2 from the Krebs cycle and transfers electrons. COQ does not pump protons. Complex 3, cytochrome BC complex, transfers electrons from reduced ubiquinol to cytochrome C, pumps 4 protons, H, into the intermembrane space. Complex 4, cytochrome C oxidase, transfers electrons from cytochrome C to molecular oxygen, reducing it to water, pumps 2 protons at ATP synthase. Complex V uses the proton motive force created by the ETC to synthesize ATP from ADP and inorganic phosphate. Proton gradient, chemiosmotic theory, proposed by Peter Mitchell. This theory explains how ATP is generated. Proton pumps create a proton gradient, electrochemical gradient, by moving protons into the intermembrane space. The energy stored in this gradient is used by ATP synthase to make ATP. ATP synthesis, chemiosmosis. Protons flow back into the matrix through ATP synthase. ATP synthase rotates, catalyzing the phosphorylation of ADP into ATP. Role of oxygen. Oxygen is the final electron acceptor, combining with electrons and protons to form water, H2O. Without oxygen, the ETC would halt, stopping ATP production. Energy yield from oxidative phosphorylation. Molecule ATP yield, NADH 2.5 ATP, FADH2 1.5 ATP. Overall, complete oxidation of one glucose molecule yields approximately 30 to 32 ATP. Importance of oxidative phosphorylation. Energy production generates most of the ATP in aerobic organisms. Heat generation, especially in brown adipose tissue, ROS, reactive oxygen species formation. Excess electrons can lead to superoxide production, causing oxidative stress. Disorders associated with mitochondrial dysfunction, mitochondrial myopathy, muscle weakness due to defective mitochondrial function, Leber's hereditary optic neuropathy. LHON affects vision due to mutations in mitochondrial DNA, Parkinson's disease, mitochondrial dysfunction linked to neuronal degeneration. Kearns-Sayre syndrome affects muscle, eye, and heart function. Thank you for watching.